Hello everyone, my name is Mrs. K. If my voice is familiar to you, hello my dear A4 peoples. If my voice is not familiar to you or is only vaguely familiar to you, that means you have Mr. K as your primary teacher or Mrs. Foye. Today, we're going over forces of evolution, changes in populations, and that is for Monday, April 13th, A Day, and Tuesday, April 14th, B Day. So this is just a reminder to make sure you are signed in to Edpuzzle just to be safe, or if it's acting funny, sign out and sign back in, and that's going to increase the chances of it working properly and submitting properly. And also, make sure you hit submit at the end of the Edpuzzle. So this is going to be similar to last time. We're going to go through our lecture and notes, so make sure you're writing notes. You'll have embedded checks for understandings and questions throughout the PowerPoint or the lecture. And then at the end, you'll have a big chunk of questions that count for your IP or your individual practice. So make sure you go all the way through the Edpuzzle. You won't be able to skip, so you have to actually watch and listen and take notes and stuff, and make sure you go through all of the questions all till the very end. All right, let's get started. So today, as our lecture title suggested, we're talking about the forces of evolution. Charles Darwin, that guy who sailed around the MS Beagle and coined the theory of evolution, only really described natural selection. Natural selection being that one trait is more favorable to another and is more prone to succeed in that environment. Something that has a trait that allows it to succeed is seen as more fit than another organism that has a trait that isn't helpful in allowing that organism to succeed and live to reproduction age. Then we've got mutations. Mutations are random changes to DNA. This is the overall driving force that creates variation, and that is what allows natural selection to act on. So a mutation happens, and because of that mutation happening, natural selection can act on it and allow things that are more fit to move on and things that are less fit to less likely move on. Then we're going to go into more detail on genetic drift. Summary of that is that random events that change allelic frequencies at random regardless of how successful they are or aren't. Again, we'll go into more of that later. And then gene flow, which is the immigration between two or more populations. Think of the ocean. The ocean flows in and out, so that allows genes to flow in and out of two or more populations. So all of the things that we described on the previous slide are forms of microevolutions. Think about what the prefix micro means, small, right? So microevolution is the change in allele frequencies that occurs over time within a population. So that's small evolution, and it occurs in short time spans. Short, relatively short, within a few generations versus many generations. Then remember macroevolution, macro prefix meaning large or big, is major evolutionary changes and the evolution of the whole taxonomic groups over long periods of time. So the creation of new species, the extinction of other species, etc. So the first evolutionary force that we're going to go into detail today is mutations. We already talked about natural selection in our previous PowerPoint lecture, so you should know that. We're going to go over mutations now. So what is a mutation? It's a change in the DNA sequence. Okay, so think back to our genetic unit, I think it was unit 6, where we talked about the different kinds of mutations. There was missense, there was nonsense, there was silent, all right? And silent, we talked about as being kind of the best type of mutation because a silent would change the code, but would ultimately code for the same amino acid being produced, which means we end up having the same protein product that we originally wanted to be produced. Then there's missense, where you aim for the target, but you miss, and you end up hitting something different, right? So the code ends up having a mutation in it, which changes the amino acid that's being produced, which then also might change the protein product. Then we have nonsense, and a nonsense mutation puts a stop to the nonsense and actually codes for a stop codon, which would change the amino acid and then possibly change the complete protein that we're having. So do all mutations affect change? No, only non-synonymous mutations do. Non meaning not, synonymous meaning same. So nonsense mutations and missense mutations are what causes noticeable mutations in an organism that may be acted on by natural selection and therefore cause a change in the population as a whole over time. Mutations are a weak force of evolution, meaning by itself evolution would be unbelievably slow, but it creates new phenotypes which natural selection can act on. So remember what kinds of things cause mutations. Mutations are caused by random spontaneous occurrence, just happens. Sometimes the DNA polymerase doesn't do its job correctly, doesn't read or write the code correctly, and that doesn't get caught, and then we end up with a mutation that carries on. Other things that could be is radiation, chemicals, or carcinogens, things like that. But mutations never occur by choice or need. You can't just decide, oh, I wish I had a lot more hair on me so I could stay warmer in winter. You either have that genetic material to create a lot of hair to help you be warm, or you don't. So where does a mutation have to occur for it to impact its offspring? So now think about cell types and cell cycles and what kinds of things get passed on to future generations. There are stomatic or body cells, and then there are germline cells. So in order for a mutation to occur and impact offspring, the mutation must occur in the germline cells. Germline meaning sperm or egg cells, okay? Because what happens in the stomatic or body cells, if you were to go out and get a tan all the time and then also pay for tanning during winter, I like to refer this to as paying for cancer, you may be causing extra radiation and extra damage to your skin cells, which may ultimately cause you in your lifetime to have skin cancer. Now, the skin cancer that you may have in your lifetime isn't going to automatically be passed to your offspring because that occurs in your stomatic or body cells. In order for you to pass something on to your offspring, it needs to be in the germline, sperm, or egg cells. So how does a mutation affect change? Well, without variation from mutations, because mutations create variations within a population, natural selection would have nothing to act on. And mutations are the random driving force behind evolution that causes new alleles and therefore new genotypes and phenotypes to show up within the population. So the image to the left shows 3 insects. The 2 top insects evolved from a common ancestor that looked a lot like the dragonfly on the bottom. So the dragonfly on the bottom had genetics that coded for 4 wings. Okay, and somehow that dragonfly ended up getting to 2 different areas or being in 2 different places, and those 2 different places had their own environmental stressors that allowed natural selection to act on our common ancestor dragonfly insect. Well, because of natural selection and time, that common ancestor ended up evolving into 2 different species, one being something that looks like a ladybug, which has a specific hind wing formation to allow it to fly, and then the other population ended up evolving into a fruit fly, which had a specific wing shape that served its own purposes that allowed it to be successful for flying in its own area. So with the mutations that occurred over time from our dragonfly-esque ancestor, it allowed us to see the evolution of 2 different species from that common ancestor. An example of mutations that impact evolution is sickle cell anemia. We talked about this when we were talking about dominant and recessive disorders and how those are passed on to future generations depending on dominant and recessive alleles. But anyways, sickle cell anemia in summary is a disorder or a disease where the blood cells take a weird sickle shape. So instead of a normal red blood cell that is almost inner tube-like shaped and carries a normal amount of hemoglobin and a normal amount of oxygen, we end up with a blood cell that is sickle shaped, doesn't have the right amount of hemoglobin, and it doesn't carry as much oxygen. So because it's not correctly shaped and it also doesn't carry as much oxygen, it tends to shorten the lifespan of individuals with this condition. They either die because their organs aren't getting enough blood, or they die because eventually they get blood clots, which can cause other kinds of issues, etc. So now you're thinking, well, if it causes blood clots and lack of oxygen, why is this something that continues to get passed on to future generations? Well, that's because in areas where sickle cell anemia was prominent, there was also malaria. Malaria was a disease that would take out individuals prior to reaching reproductive age. Individuals with sickle cell disease had a resilience against malaria. So instead of individuals dying prior to reproductive age because of malaria, they were able to live past reproductive age, passing on the trait of sickle cell, but then ultimately they still have a lesser lifespan than a non-sickle celled individual, but because it allowed them to move past reproductive age, the trait is seen over and over again. So this kind of impacts evolution on a slow level amongst different populations of humans. Another example is antibiotic-resistant bacteria. So around the 1940s, scientists with antibiotics eradicated a lot of health threats made by bacteria. It was seen as a type of miracle drug, and people were taking it for all sorts of ailments, even colds. So basically anyone would go into a doctor with a sniffle, and the doctor would just throw antibiotics at them. But we know now that, we know that colds are a virus, not a bacteria. So throwing antibiotics at the common cold isn't going to be helped, and the cold would end up going away on its own in a few days anyways. However, in the past 2 decades, we've seen a resurgence of infectious diseases as well as brand new ones. And this is because when antibiotics are used, there's always a chance that some of the bacteria will have some sort of random mutation that lets it be resistant to the antibiotics, which leaving those strains to survive and live and pass on their genetic information. So the antibiotic-resistant bacteria can live on and pass on its information, creating more resistant bacteria. So combine that resistance with people who don't finish their entire prescription of antibiotics. Always make sure you're finishing your entire prescription. And combine that with the possible unnecessary use of antibiotics on livestock, and we can be unintentionally creating superbugs or super resistant bacteria. So this is why we're seeing nowadays reductions in antibiotics in agriculture. Now it's reactive usage of antibiotics versus proactive uses of antibiotics in agriculture. And doctors are now not just throwing antibiotics at everyone who walks in. Now they're asking a lot more questions, making sure you've been miserable for at least more than 7 days to show that it's not a cold and it's actually probably a bacterial-related illness. And that's helping us keep the bacteria that are currently working, hopefully working longer. So this image below shows, a culture of bacteria before antibiotics. They had a varying level of resistance, the lighter or more yellow colors being very little resistance to antibiotics, and the oranger and redder colors having a higher resistance level. So this is a culture, and you have a varying type of phenotype showing up on the culture. After antibiotics, only the most resistant bacteria are still around, and therefore the final population overall will end up with a higher resistance level than before the antibiotics. So the next thing that we're talking about after mutation is gene flow. Gene flow is the migration or movement of genes from one population to another. Genes can move back and forth between populations. So in the image below, we're seeing that a population of brown beetles and a population of green beetles. An individual from the brown beetle colony moves over to the green beetle colony and breeds with them. This is going to change the allelic frequencies, so we might start seeing more brown beetles showing up in the green beetle colony, and if those beetles were to move back to the brown beetle colony, we might start seeing some green beetles showing up. An example of gene flow, there is a scenario where there's a species of snakes on a continent or a mainland. This population of snakes has a genetic variation naturally to be light brown and dark brown, almost black. So just like people have the genetic variation to be very pale or very deep toned, this species of snake has the genetic variation to be, light brown to very, very dark brown, almost black. On the continent and on the mainland, the soil is rich and fertile and dark. So the majority of the snake population there is dark brown because they blend in easier with the dark soil and the bird of prey cannot see them as easily. But of course, there's still some light individuals, but natural selection selects against those, okay? But one day a big pile of driftwood comes to shore and has a lot of yummy bugs on it. So the small groups of snakes go into the driftwood to eat. Unknowingly, the tide comes in and picks up the driftwood and carries it to a nearby island. On this island, there is a lot of sand and the soil is poor and light brown colored. So all of the individuals that were on the driftwood, they started having their own breeding populations there. But because the soil is a different color, the lighter brown snakes ended up having a better survival rate and a better job at avoiding birds of prey. So over time, the population on the island became more light brown than dark. However, there is still a high occurrence of driftwood floating from island to mainland, so the gene flow and its gene variation still allows the two populations to be considered one. They could still interbreed with each other, they could still share their genetics. It's just due to natural selection, the snakes that tend to survive on the island are lighter colors, and the snakes that tend to survive on the mainland are darker colors. If for some reason some big giant earthquake or volcano were to occur and a giant mountain range were to somehow form between these two populations, they might be separated long enough for natural selection to occur and turn them into two distinct species. But in this scenario, because there's driftwood going back and forth, they're still considered one species. So genetic drift occurs from random events that can alter allelic frequencies regardless of what the traits are. Okay, so some random events could be hurricanes, tornadoes, earthquakes, human interference. So we could do some overdevelopment and destroy habitat, things like that can cause changes in allelic frequencies regardless to what the original traits were. There are different kinds of genetic drift. Again, genetic drift being different kinds of allelic frequencies showing up in the population. And one of the ones that we're going to talk about is the founder effect, which is taking a small sample of a population and founding or starting a new population from it. So in the image below, we have an original population of 20 blue individuals and 10 red. So in this species, they have the genetic variation to have blue and red individuals. But in this population, it appears that blue is more successful, and that's why there are more blue individuals. However, there is a small subset of that population that gets put into a new area to start a new population from it. In this scenario, the new population is mostly red and little blue. So think about this. Is this new population representative of the original population? Think about it for a second. No. If you answered no, you're correct. The original population was mostly blue with a little bit of red, and the new population is mostly red with a little bit of blue. And because it's not representative of the new of the original population, we're going to be seeing different phenotypic frequencies because there is more red phenotypes to share their genes. The blue phenotypes and genotypes are still there and will continue to show up in the population, but if the selective forces and environmental stressors in both populations areas are the same or very similar, we might end up seeing ultimately the same proportions over time. Mostly blue in the original population, and we might end up seeing mostly blue in the new population if the selective forces and environmental pressures are the same. But the proportion might remain different if there's some kind of selective pressures that select against blue in the new area. And if there's a scenario where this is a complete isolation from the original population, which would remove gene flow so they can't breed back with the original population, which then maybe the blue would be removed from the population as a whole, and other selective forces may eventually change the population so much that we would see just red individuals. Those red individuals will also change and adapt to their environment, and the species or the population as a whole will change so much that they would not be able to interbreed with the original population, and therefore a new species has formed. So that's pretty cool. Then we have the founder effect. So it's another kind of gene flow. The founder effect is the loss of genetic variation that occurs when a new population is established by a very small number of individuals from a large population. So on the last slide, we took some individuals from a large population and put them in a new area. Those individuals will then have less of a genetic variation than the original population because they could only breed amongst themselves and not have much new gene flow coming in to increase variation. The founder effect is often accompanied with a genetic bottleneck. We'll talk more about that later. So the genetic bottleneck, the image on the left, shows literally a bottle full of marbles, blue and yellow marbles. Because of the shape of the bottle, only a certain number of individuals can pour out very slowly. So the parent population looks about 50-50 yellow and blue. We pour out some of that parent population into a glass, and what's in there will end up being a drastic reduction in population, and then the surviving individuals will then go to fill up their own bottle, but because the ratio of individuals that were put in the glass was different than the original ratio of phenotypes in the parent population, the next generations of the surviving individuals might be at a different phenotypic frequency than the original population. So the bottleneck effect could be from when a lot of individuals die off or are physically moved by human intervention, and the few that remain do not represent the old population. Again, their phenotypic frequencies are different than the original population. And this could be due to natural disasters like hurricanes, earthquakes, floods, human intervention from overhunting or overdevelopment, etc. So an example of genetic drift with the founder effect as well as with the bottleneck effect would be the white-tailed deer. So in the 1920s, deers were overhunted. They were overhunted to the point where they were almost extinct, and they were actually declared extinct in some states like Kansas and even Indiana. So protections and hunting limits were placed on them to recover the population, and clearly it was very successful as we have white-tailed deer all across the country. But in some areas where the deer were eradicated, like Indiana, new populations were needed to be added. So they went to Wisconsin, they went to Michigan, they went to Virginia, and they collect random subsets of those populations where deer existed and drop them off in Indiana to start a new population. This took, again, a small subset of the entire species genetics and put them in the new area for them to grow amongst themselves until their territories overlapped or migration occurred. When territories overlapped or migration occurred, that allowed gene flow between populations to happen. And gene flow between various populations keeps a species happy and healthy and genetically diverse. Okay, they've got a big bag of tricks. The genetically diverse thing allows them to be able to adapt and succeed in case of catastrophic events. However, there are some areas where the gene flow did not occur. So an example would be military bases. So in military bases, deer were added, but because of the super tall fences that are found there, gene flow was not able to occur with the outside populations. The deer could not jump the fence to leave and breed with the outside populations, and the outside populations were not able to jump the fence and breed with the inside population. This caused the deer to have a founder effect. So just the few individuals that were added to the military base constantly bred amongst themselves with no new genes from outside. And this ultimately can cause detrimental traits to become more prominent, or the population to have less variability if or when events occurred that would require certain traits to be more fit. Okay, so they're kind of just inbreeding amongst themselves. But within the military base, once the population recovered and was becoming overpopulated, they enacted a hunting season. So the hunting season ultimately cut the deer population in half every year. So not only does this population have a founder effect of already having restricted genes because of just the few individuals that they came in with, but now that there's hunting and the population is cut in half every year, this is now going to create an additional genetic bottleneck where the already low genetic diversity of the population was becoming 50% lower each year. So the random deer that survived would then pass down their genes, and this would ultimately kind of affect the population's success in the future because they're just losing more and more genetic variation every hunting year. Okay, which isn't great. So this could easily kind of be reversed if the military base brought in new deer or allowed these deer to leave or cut down holes in the fences. But on this specific base that I'm talking about, we're kind of doing an experiment to see how genetic bottle effects, along with the founder effect, is affecting this population. So it's kind of just an experimental area. So to a more fun fact kind of thing though, if you're a hunter or know people who do hunt, especially deer, you are aware that hunters have kind of an obsession with bucks and large racks. All right, so the bigger the antlers, the more points you get, and the cooler of a hunter you are. Okay, there's actually a reason why we have this kind of obsession. So during the repopulation efforts, biologists knew does were important, does being the female deer, doe, a deer, a female deer, blah, 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 blah. Okay, biologists knew does were important to the growth of the population, but because deer were a means for some people to survive, they couldn't completely eradicate hunting. So they encouraged the hunting of only bucks because they knew that you only need 1 or 2 boys to impregnate a whole herd of does, okay? And because of this, and this embedding of the idea of only hunting bucks, it somehow morphed into something I like to call antler porn, which is where hunters compete to get the bucks with the biggest rack of antlers worth the highest point value. So because of our repopulation efforts, it kind of changed the mindsets of hunters to avoid does and instead fixate on bucks, and to make it more fun, fixate on bucks with big racks, okay? But now that the population has recovered, this is kind of actually detrimental now. We're starting to see overpopulation amongst our deer groups, and therefore if we were to kind of shift our mentality to maybe $2 and a doe, we would overall drop our deer population to a point where they could all eat happy and healthy instead of all of them being kind of skinny because there's too many deer. So if you're a hunter out there, does taste good too. All right. Well, this concludes today's lecture portion of this. I'm going to pause it after this, and you'll have an IP portion. So make sure you're going through all of the IP and getting through all of the questions and hitting submit at the end. If you have any questions, Mr. K and I have our office hours posted on Haiku. So does Mrs. Foye. So please come visit us in office hours, ask us questions, etc. Or you could just send us an email and we'll get back to you as quickly as possible. All right, well, that's it for today's lecture. Have a great evening. See ya.